Hello and welcome. My name is Matthew Greer. Today I will be presenting on G protein-coupled receptors. This is presented through Chemistry 530, Principles of Biochemistry, through Grand Canyon University. My professor is Dr. Karma Cook, and today's date is the 8th of June, 2022. If you've never talked about G protein-coupled receptors before, this is the perfect place to start. This is an exciting group of proteins that are very active in the human body and very active in all eukaryotes across the board. This is what they look like. They are this structure here. This is the cell membrane. They sit in the middle of it. If you aren't familiar with these structures, don't worry. We're going to talk about each one of these chunks. We're going to break it down. We're going to talk about the anatomy of these proteins, and then we're going to be talking about their physiology after that. So let's jump on in and talk about what we're going to learn during this presentation. So let's start on what our objectives are for this presentation. We are going to take a look at what are the GPCRs? What are G protein-coupled receptors? What are they? What do they do? What are the different types of GPCRs? We will look at the basic types of them, go over some examples of where those may be used. We're going to talk about the structure of them. We're going to break apart the different parts of a GPCR and show how those help that molecule have the function that it does. We're going to talk about how G protein-coupled receptors are activated and then how they are inactivated. And we're going to be using epinephrine as an excellent example of this. And then we're going to talk about an example of what happens when GPCRs are implicated in disease. And we're going to specifically talk about the cholera and cholera vibrio. Before we get too crazy and we jump right into GPCRs, let's just real quickly talk about a couple of biochemistry background, make sure that we're solid on all of this before we jump into the new stuff. So just as a review, a receptor, this is a specific binding site for a certain molecule or environmental stimulus. Receptors can look very different. They can be light molecules, they can be hormones, they can be fats or sugars or other kinds of molecules. They can be all kinds of things. Enzymes is a substance that brings along a biological change. Most of the time, these enzymes are made out of proteins. The substrate is a material on which an enzyme acts. This is the enzyme that is having that biological change from the substrate is the material that is having that biological change from the enzyme. A conformational change is a shift in the three-dimensional structure of a molecule. For this purpose, we'll be mostly looking at proteins, but it can be the three-dimensional structure of change of all kinds of molecules, sugars and nucleic acids. And then the last kind of touching point, and this is the important one and what the majority of this presentation is going to be about, is signal transduction. And this is the process and the kind of important series of events that happens where a cell responds to substances outside of the cell or in the extracellular matrix, and through signaling molecules that are found on its surface, relays that signal inside and then affects some kind of change, whether it's metabolic or whether it's transcription factors. Some kind of change is happening inside of the cell in response to this external stimulus. Okay, now that we have kind of reviewed a little bit some of the basics of biochem, getting into this discussion, let's jump in and talk about G protein-coupled receptors. What are they? They are a diverse group of membrane receptors in eukaryotes. They're present in all eukaryotes. They're in animals, they're in plants, they're in fungus, they're in protists. In humans, there is close to 1,000 in humans. And they're very important molecules because they give a lot of information, like we said, to the cells about their external environment. So they can receive signals from light, from peptides, sugars, proteins, and they give information about the environment, or they can be used to send information inside of the body. An example is yeast will use GPCRs to sense their glucose levels, and in plants, they're used in the process of opening and closing their stomata. Like we said, there's 1,000 GPCRs in humans, and right now on the market, about 30 to 40% of our marketed drugs are targeting these G protein-coupled receptors. So they're an important group of molecules that have a high amount of interest to us. All right, let's talk about the different types of G protein-coupled receptors and their uses. In vertebrates, there are 5 main categories. There's class A, B, C, the frizzled family, and the adhesion family. And like we've touched on before, there's a lot of different things that can be ligands for these receptors. While all these receptors have the same basic 7 helical structures through here, they all have the basic binding on the outside, interaction on the inside. There's lots of different signaling transduction that happens. This is the largest group of signal transduction proteins in the body. Class A, rhodopsin, is the largest. It is the most diverse. It includes things like vision. It includes things like smell, taste. The other ones like secretin are involved in some glycogen breakdown. There's adhesion that helps cells adhere to the extracellular matrix and things that are going on out there. These are a very diverse group. Like we said, there's a lot of drugs that are being formulated to kind of help bind to these and have mediated processes. But before we get too technical and before we get too much into the brass tacks, let's talk about some structure of these proteins, and then we can get into a little bit more of their function. Okay, now that we've talked about the different classes of G protein receptors, let's jump into the structure of these guys. This protein is a single polypeptide, meaning it's just one chain of amino acids. Its three-dimensional structure is what this image is showing here. And it's got 7 transmembrane loops. And there's these 1, 2, 3, 4. They alternate. Loops going through the membrane here. Transmembrane means that it goes through the membrane and is embedded inside of it. There's lots of hydrophobic chains on these amino acids. These R chains are a lot of hydrophobic structures. This really binds that molecule into the membrane and prevents it from moving around. Because there are 7 transmembrane receptors, we can also refer to these guys as a heptahelical structure. On the outside of the membrane, in the extracellular space, there is an amino group here. And on the inside of the structure here, there is a carboxyl group. Now these groups are polar, and they are the parts that are going to interact with the amine group is going to interact with things that are coming in the extracellular matrix, and the carboxyl group is going to interact with things that are coming from the intracellular side. So most times the signal will be originating from the external side of the cell, the external matrix. It'll be binding up here, and then the signal will be sent through this membrane to the internal side here, where some effect will take place. All right, so the structure of the actual G protein, and this is the internal part here, that is on the intracellular side here in the cytoplasm, on the interacting with the carboxyl terminus here. It is what we call a heterotrimeric protein, and it consists of an alpha, a beta, and a gamma subunit. And these are the alpha and the gamma are anchored inside of the plasma membrane, and this beta is inside of the cytoplasm itself. The purpose of the G protein is to bind GDP and GTP. And remember that GDP and GTP are guanosine nucleotide derivatives with guanosine diphosphate and guanosine triphosphate. They have that triphosphate has that additional phosphate group, but G proteins are themselves guanosine nucleotide binding proteins. In most of our G proteins, when the GDP is bound, the protein is inactive, and when the GTP is bound, that triphosphate, the protein will then activate and be able to do whatever function that it's designed to do. So when we say the G protein becomes activated, what we're meaning is that at this point in time, the G protein has some ligand or some molecule that is binding to the outside side, to that amine terminus out there. And when this binding happens, we get a conformational change inside of the 7 transmembrane units, where these units are going to readjust themselves in such a way that the G alpha here, this alpha subunit of the G protein, is able to bind this GTP. And the example that we're going to use is an agonist. It is epinephrine. When an epinephrine molecule binds to the outside of this GPCR, it makes this conformational change where this ADP is traded for an ATP. When this happens, the G protein will cleave. It will split into two parts. It will split into a GTP-bound alpha subunit, which is what we're going to talk about is the main effector here, and then it will also split into a beta-gamma dimer. Whereas this molecule of beta and gamma together, it's still going to remain, both of these are going to remain inside of the membrane still. But since they are separated, they're now able to transmit signals, they're able to activate other proteins. And while we're going to focus on the G alpha and the GDP-GTP transmission and transduction, the beta gamma dimer is also able to carry out signals and affect other tissues and other processes in the cell. All right, so G protein-coupled receptors are usually called G protein-coupled receptors with second messengers. And the first messenger is whatever is binding on the extracellular side. So in this case, we're talking about epinephrine. This is the first messenger. It is going to bind on the outside, the extracellular matrix of the G protein. It makes the conformational change that allows the G alpha to become activated. The activated G protein will then activate another protein into what we call a second messenger. There are some different methods that G proteins can do once they become activated. They can stimulate, which is what we're going to focus on, or they can inhibit activation. We're going to talk about adenyl cyclase, and G proteins can both stimulate or inhibit this adenyl cyclase. So in this case, the adenyl cyclase, after this GDP is switched to GTP, after we bind that, this G protein, which is now activated, can bind here. This is going to activate adenyl cyclase and converting ATP into cyclic AMP. And cyclic AMP is a great second messenger system. It is able to do a lot of things. Signals that use cyclic AMP as their secondary messenger include corticotropin, follicle-stimulating hormone, glucagon, histamine, luteinizing hormone, other ones like parathyroid hormone, serotonin, and some tastants like bitter and sweet tastes also use the cyclic AMP as a secondary messenger. Now, cyclic AMP, once again, this is a cascade. This is a signal transduction cascade. So it is cyclic AMP will go and it will, as a second messenger, activate protein kinase A. And protein kinase A will go and do a whole bunch of other cellular responses. It will go and affect a whole bunch of other proteins and have all kinds of mediated cell responses that it will do, whether it's opening ion channels or doing transcription factors. There is lots of things that can happen once this protein kinase A is activated. And this process will continue as long as this activated G protein is bound to this adenyl cyclase. It will continue to turn ATP into cyclic AMP, which will go to protein kinase A, which will affect more cellular responses. So it will just keep going until it's eventually stopped. So how is this process of this constant activation of pKa ceased? Well, the inactivation of this stimulatory G alpha unit is self-inhibitory. So what this means is eventually it will convert this GTP back to GDP, and at this point in time, the alpha subunit will dissociate from adenyl cyclase. And at this point in time, it will reconnect with the beta and gamma subunits that are nearby, and they will bind back together, and those will bind back to the GPCR, the G protein-coupled receptor, and at this point in time, the process is able to begin again. If that another first primary messenger comes and binds to the outside of the GPCR, it will affect the whole cascade over again. And pKa will once again start being produced after it's been activated by CAMP, which has been activated by the adenyl cyclase. Earlier we talked about the fact that normally the activated G protein and GTP will normally split themselves apart, and they will go back to that inactivated state and be ready for another agonist to come and bind. But what happens if that doesn't happen? Let's say epinephrine stays bound here. Well, luckily, there is a couple of ways to desensitize or create kind of a weaker link here and get that GPCR ready to once again accept another signal. So in the case that a ligand stays around, and let's use epinephrine as an example again, beta-adrenergic receptor kinase, or beta-ARK, will phosphorylate amino acid residues. And these amino acid residues are on the carboxyl terminus of the GPCR. So they are on the end that binds to the G protein. And beta ARK will also sometimes be wrote GRK, which GRK stands for G Protein-Coupled Kinases, receptor-coupled kinases. And this GRK will phosphorylate, it's the serine amino acids, for those of you that are interested, around this carboxyl end here, and it allows for the binding of beta-arrestin. And beta ARK or GRK is activated by increased levels of pKa. So as this agonist continues to bind, it continues to make that G alpha active with GTP there. It will continue to make more CAMP, which continues to activate more pKa. So this increased pKa now activates another protein in kind of a negative feedback loop here that will shut down this process. So as beta-arrestin comes in and binds, it blocks the G protein interaction. This blockage of this interaction here allows for the removal of this agonist. And then this beta-arrestin will get recycled, usually through endosomes. And then at the end of this process, the GPCR, the G protein-coupled receptors, will be able to bind another agonist. So the purpose of this pathway is to, in case this switch gets stuck in the on position, there is an ability for the body to remove this kind of situation. Now we're going to talk about, coming up next, a kind of unique method here where we have a disease process come in that is turning this switch on and keeping it on. And we're going to be talking about the cholera bacterium and its kind of effect on this pathway. But beta-arrestin and beta-ARK are really important for the desensitization of these molecules so that they can reset to normal, because too much of anything is bad and the cells don't want to be turned on all the time. And we'll talk about why that is coming up right next. So now that we've talked about normal G protein-coupled receptor function, I always like to explore and look at what happens when a problem happens in the body, when we have some kind of pathology or disease process, whether it's genetic or some kind of organism that is hijacking the body system. And I like to look at how that affects the normal process. So in normal gastrointestinal epithelial cells, chlorine is excreted through the cystic fibrosis transmembrane conductance regulator, which is CFTR. And this occurs through a GPCR cascade involving the activation of pKa, like we have discussed previously in the last couple slides. So Vibrio cholera produces a toxin that locks the G alpha into the active state. And cholera is a well-known disease. It's been around for a long time. This toxin that produces has the effect on the body where it creates large amounts of watery stool. And in patients that don't receive any treatment, there is rapid dehydration, as there's a lot of hypovolemic shock and individuals lose so much water that they become metabolically acidotic. Now, cholera isn't everywhere in the world, but it really is common, and more common in places that have unsatisfactory hygiene or areas that have natural disasters like tsunamis or floods, or anywhere that you have seawater and freshwater mixing. Luckily, if patients have treatment, 1% of them is fatal, 1% of cases. But it's transmitted through contaminated water and food. We are the only natural host for cholera. It doesn't make any other organisms sick. However, this bacteria can live inside a shellfish. So occasionally around the world, we will see outbreaks of cholera in shellfish that have been holding on to this cholera, and this cholera has been living in there. So once cholera is in the human system, we said it produces a toxin. And this toxin goes through the membrane and it locks the G protein into the active state. So this G protein is constantly having that adenyl cyclase pump out cyclic AMP. We're taking ATP, we're turning it into CAMP over and over and over and over again. And this activates pKa, which activates a whole bunch of things, but what's really important in this disease process is the activation of that cystic fibrosis transmembrane conductance regulator. And it's called that because it was originally, it's a factor in cystic fibrosis. This protein does not function properly. In this case, what happens is it continuously pumps chlorine out of the cell. Now, chlorine is 1/2 of salt. Usually, wherever there's chlorine, there's also sodium that is going to follow it. So as chlorine is pumped out of these epithelial cells, the sodium is going to follow it out as well. And as the chlorine and sodium continue to flow outside of the cell, water is going to follow those solutes through osmotic pressure. So this is why we get the really, really profuse diarrhea. As more and more chlorine and more salt are removed, that water is going to follow it. This is also why patients can't just drink a whole bunch of water to get over having cholera, because they're losing so much salt through losing both sodium and chlorine. Usually when we do treatment in these patients, they will have to have saline fluid as well. So you can't just orally take water to get over cholera. You have to take saline solution that also helps to get your salt levels back up, to replace this sodium and to replace this chlorine that's happening because this GPCR is just constantly activated. So while normally this process would shut itself off and this activated G protein would deactivate itself and rebind to the GPCR, because of the Vibrio cholera toxin, it is unable to do so. So it just continues to pump out this chlorine and sodium. This concludes our introductory kind of jaunt into GPCRs. There's a lot more to learn about these, but I hope that this gives you the basics, that you can, after watching this, you know what GPCRs are, G protein-coupled receptors, you know what they are, you know what the different types are, you have at least an introduction to the different types of them. You can see what their basic structure is. That's most of the 1,000 or so GPCRs that are in humans have the same basic structures as the ones we've looked at. You can see how they're activated, inactivated. I hope that you have a kind of appreciation of what can happen now when disease is implicated in GPCRs, such as cholera. Something important to note is other diseases such as allergies, depression, blindness, deafness, diabetes, and some cardiovascular defects also have implications that are GPCR related. The references that I used here to create this slideshow are listed. If you want to go and learn more about the cholera and how we're actually treating that, this Thomas Ramada Naran study is really good. I suggest it's a really good read. If you want to look at just the basic structure and function, either this Rosenbaum and Nelson, and if you want to look at these, they're working to create a really large collaboration on the different types of GPCRs in humans. This one in Nature Reviews is really good by Stevens. I hope that you enjoyed this presentation, and I look forward to seeing you in the next one.